Good morning, everybody. Could you please let me know if you can hear me well? Great. Very good, very good. Um, so how is it going, guys, with you with this course? Everything is going okay? Well, I'm recording right now, but this discussion I can trim it out if somebody is not interested to have that recorded. I'll give you just a few minutes if you have any comments regarding the previous lectures. If there's any material that is not really clear, anybody has some speculations, questions, comments?
Hi, good morning, Professor.
Good morning.
I was wondering if you could go over the Tata box, if you could just re-explain it real quick.
The data box.
Yeah.
Okay. Any other requests? Okay, so nothing really except that question. So uh, in general, just regard a general point about the data box. So maybe speak to disease post-translation modification example briefly. Yes, Margaret. So you can see that post-translation modifications, as I said, those are in almost every single disease we are going to discuss. And you will see today that we will come back to discuss some of those again and again. So because the regulations of proteins in terms of function take place mostly at the post-translation modifications. So after this lecture, if you think you need more examples, let me know. But I have a lot of examples today. Okay. So guys, please, because when I'm talking, most probably I will not be looking very carefully for the chat. Feel free to turn on your mic and speak if you have something and you see that I'm moving forward fast, right? Now, just to answer your question about the data box. So I mentioned that for the transcription to take place, there must be certain steps. The first and the most important step is to find out where would be the transcription initiation. And for that, we would need consensus sequences, and those sequences, some of them, one is the most famous, is the Tata box, where we would know, like the RNA polymerase would recognize those sequences as potential sequences where it can bind and initiate the transcription process. Of course, it wouldn't do this on its own. There is a complex of multiple proteins that play a role in that. So there will be some sort of scanning that is taking place on the promoter region upstream of the gene to recognize where is the data box, and then the transcription machinery will start binding to that and then initiate the transcription process. Okay. So as for today, we will kind of finish up with most of the molecular aspects at the level of the DNA. We discussed in the previous class the protein generation, the protein modifications, and how that would affect a protein function. And then we described how proteins are made in the previous lecture. So we described how the transcriptional machinery is initiated and what are the modifications that could take place during the RNA processing. After that, we discussed the generation of the RNA that has been spliced, so generating more than one protein from this the same RNA, plus the modifications that we need to have in order to stabilize the messenger RNA so that we could ensure that this gene transcription leads to protein translation. Today, we will go through the genetic material replication, the DNA replication. We will discuss a bit about the mutations and how these mutations could lead to certain diseases. And we will briefly discuss about genetic diseases. So now we will start kind of step back and say, what are the diseases? And we will focus on one of these. So today we'll kind of focus on the genetic diseases. But you can see throughout the presentation that this is really a rough description, because genetic disease at the end of the day probably could lead to cancer. So the definition doesn't mean that it's only related to genetics. Again, I just want to emphasize that we still have office hours. I had only probably 2 people that showed up last week. And guys, you really need to go through the material, don't accumulate it. Please, if you have any questions, come as soon as you can so that you are back on the track. Now, as for today, we already covered most of the molecular aspects of proteins, lipids, carbohydrates, DNA and RNA, how they are generated and how do they interact. We will discuss today protein DNA interaction, because this is a process that takes place at the level of the RNA replication. Also in the previous lecture, we discussed intensively the communication between cells and how it takes place. So today we will cover the second aspect of a cellular function, which is how the cell reproduce. You can see that with time, we are covering more and more of these aspects. And we are kind of trying to relate the molecular aspects to the cellular function. So we discuss about the protein phosphorylation, how it affects a cellular communication, or how DNA damage could affect the reproduction of a cell. So it's merged between the two, and that's why I can't go really very, very deep on most of the aspects. So I kind of keep it a little shallow. I just want to warn you today that there are a few proteins that you want to know their names and their function, because if you don't, it's going to be very tough for you to retain the information in the future. Okay, so what's the learning objectives of this class and the next class is going to be understanding the cellular reproduction, which is the cell cycle, how the cell prepare itself in order to divide, and how the cell will build the material that is checked to be accurate before proceeding in the cell cycle and the division. Part of that is going to be DNA replication. And once DNA replication takes place, then the cell is ready to divide. If this is a somatic cell, it's going to be dividing through mitosis. And if it's a gamete or an egg, it's a sperm or an egg, it will be dividing through meiosis. And we will discuss the mutations and how these mutations could rise and what would the cell react toward that. So there will be some discussion about the DNA damage response. And probably we will not get into the level that we discussed the types of cell death, because the types of cell death are dictated by many of these processes. So probably the introduction of next lecture would be this cell death. I just want to warn you, I'm sharing my screen. So please, if I'm talking about something and you don't see it on the screen, warn me directly because this happens a lot on online classes that I'm talking, for example, about the cell cycle and I'm pointing to the cell cycle. However, you don't see that. So obviously you don't see the material, then you will get lost. Okay. So as I said, we are going to cover in this class 6 different categories of diseases. Today is going to be the genetic diseases. The next one will be the metabolic diseases. And as we go through the list, we will always prepare an introduction about the type of the disease. For example, in the metabolic diseases, definitely we will have to discuss about how the gene that are responsible for the generation of the ATP and the energy in the cell. And definitely we have to discuss about the cellular biology, about the organelle, which is responsible for that. That is the mitochondria. And then we will discuss the shift from the steady state to the disease state. So how can we define genetic diseases? Simply put, these genetic diseases are defined by the presence of an abnormality in the genome. And the abnormalities in the genome could be something that is very simple, like mutations are defined by a change in the DNA sequence of a gene. Now, it's very simple. However, depends on how important that gene, it can cause lots of troubles. And how much that gene in terms of function, how much function it regulates. You will see today, for example, the protein P53 or TP53, which is a tumor suppressor, it can regulate up to 500 genes. So you can imagine that a mutation that takes place at the level of a certain gene could affect 500 downstream targets or more. So this abnormality can range from a single base change in the DNA sequence that could happen at the level of the replication, and it's carried over. And it could be possible that the abnormality at the level of the chromosome itself. So you would have a change of a small chunk of the chromosome. So it's an addition of a small part of a chromosome to another, or a subtraction. So you lose part of that chromosome, or you could lose the entire chromosome. So the genetic diseases range from that single mutation to the loss of the entire chromosome. Now, definitely, when we say genetics, we have to remember that some of these genetic diseases are carried over from previous generations. So some of the genetic disorders are inherited by parents to the offsprings. And these, most of the time, we have the genetic tools to detect them. And this is what happened during pregnancy, that the doctors most of the time would suggest on the mother to perform some of the genetic tests to check the predisposition of certain genetic diseases that are known. While this is the classical definition of a genetic disease, of course, genetic diseases can happen by acquiring them through changes. And those changes can cause mutations, and therefore it doesn't need to be inherited from the previous generation. Think about the UV light, how it can cause damage in the DNA. And the more damage it can do, probably it can lead to some of those DNA not repaired correctly, and therefore the cells could form like skin cancer. Some other aspects of the genetic instability comes from merging more than one genome together. Recall in the last lecture, we discussed the life cycle of the viruses. So one of the steps of the life cycle of the viruses, this is one of the acquired changes that might happen at any time, that a virus infects our cells, and then the virus needs to reproduce itself, so it has to recruit its genetic material into our genome. And if this takes place, then uh, some of our genes might need disrupted or affected. And therefore, this is a change that can continue throughout life. So this is an acquired change compared to an inherited change. So mutation could happen randomly or due to some environmental exposure. Like, as I said, there's known chemicals like carcinogenics that cause the mutations of the DNA. UV light is known to make DNA breaks that could probably lead to some mutations down the road. So here I listed for you some of the examples. We discussed before Huntington disease, where we pointed out that it is only a phosphorylation event that happen at the Huntington gene that causes this brain disorder. But it could be related to a mutation that causes the Huntington gene to be phosphorylated or not. The most famous, and one of the most famous, it's not the most, one of the most famous is the cystic fibrosis. And this is a mutation that takes place in the transmembrane protein, the CFTR. We as biologists, we always like to make contractions of the names to make things very difficult for you guys. And when this protein, because it's a transmembrane protein, when it's mutated, then it will lead to an improper localization of the membrane into the protein into membranes. And therefore, it will lose its function as a chloride channel. Now, losing the function as a chloride channel, meaning that there is an osmotic imbalance, so that there is a change in the amount of the water and mucus that is transferred across the membrane, and therefore these mutations lead to the thickening and the buildup of mucus in the lungs. And definitely when you speak about congestion in the lungs, that is lethal. And therefore, because it will make the exchange of oxygen carbon dioxide more difficult. So this is one of an example that it's a simple mutation that takes place in the CFTR gene, could definitely lead to cystic fibrosis, and it affects multiple tissues, as you can see in lungs and in digestive tract. We will focus on cancer in the next probably 5-6 slides, because it's very well studied and it's easy to cover. Also, I have material for you added as PDF. I'm not sure if it's visible yet for you on Brightspace or not, but I have few pages of PDF. If you want to read more about some of these diseases, you can go for it. The mutation that causes the sickle cell anemia is also very well known, because that is a systemic disease that causes the exchange of carbon dioxide, oxygen more difficult. And this is not only not the only complication. The other complication is that these sickle cell shaped blood cell will obstruct the blood flow. And this is probably the most problematic in causing the in hypertension conditions, in the blocking of the blood flow, and in ischemia reperfusion aspects. Okay, so let's start by looking at the cellular reproduction. Now, obviously, I'm taking my time to describe the material easy on you, so that you understand what we are discussing, and probably what is the aspect that could come out from that. And then you can see that probably now you know the style. So most of the time I go forward, I add a little bit on the concept, and then I go back and give an application. So let's discuss the cellular reproduction and what are the steps of cellular reproduction, and where would be the points we have to discuss in terms of the post-translation modification of proteins that could cause a disease, and what are the DNA modifications that could cause to a gene shut down on or turn on. So the homeostatic uh, balance in our body maintains a subject where we have a number of cells that are functional, and obviously these cells will not survive most of the life except in certain tissues. So these cells need to be dividing at certain point. Obviously, there are certain cells that do not divide, like cardiomyocytes in the heart, neurons in the brain, and there is very little regeneration that takes place there. But in most of our body, the cells don't last for long, and some cells last for a certain period of time, and therefore there must be a process where these cells produce the material needed in order to divide into two exact copies, and this process has to be tightly regulated. So in most tissues, new cells are created as fast as the old ones die. So that's how the balance between cell survival and cell death maintained to keep the homeostasis of our body, that there will be always cells being functional and perform the function they are supposed to. So this is a necessary process for the life maintenance. Now, the reproduction of our cells is divided into two parts. As I said, if it's a somatic cell, this means it's not an egg or a sperm. And this is describing eukaryotic cells, just to make it clear. It involves the division of the cell. First, it will be the mitotic process, which is the nuclear division. And when we speak about mitotic division or mitosis, this means the cell has to create two copies of the DNA that are exactly the same, and they are exactly correct, and there will be no mutations or mismatches. And at the end of this process, the cytoplasm divide, which is the cytokinesis. Okay, now as you can see on this scheme, this is a very tightly regulated process. So cellular reproduction passes to two checkpoints. One checkpoint is when the cell being preparing the material for the synthesis of the DNA and proteins, and one after that process. So before a cell can divide, what we said is it must double its mass and duplicate its content, all the content. Now, the preparation of this division happens in what we call the interface here. And that interface has the first checkpoint during the preparation of those material, and then after the synthesis of those material. Now, the alternation between the mitosis interface in all tissue with the cellular turnover this is what we call the cell cycle. Like, we have to pass all the steps one by one and pass the checkpoints one by one in order to proceed. The best decision a cell can take if there's an issue is, what's going on here? Yeah, the best decision the cell can take if it's going through the cellular reproduction and there's a problem, then it goes to cell death. The worst situation is that carries that mutation or damage to the next generation or the next cell. So the four phases are the S, which is the synthesis, where the DNA is mostly synthesized within the nucleus. Then if we follow up, then there's the gap phase, where the RNA and proteins being synthesized. And this is the period where we will have the completion of the DNA synthesis. And we have the checkup on the DNA material that it's ready to go for the division process. If it passes through this checkpoint here that you see in the previous slide, if it passes through, so we synthesize the material, we duplicate the DNA, and then we pass through this checkpoint here. If this is true, then the cell will go through the mitosis and it will divide the nuclear and the cytoplasmic material. Once that happened, you could call G1 phase as a recovery phase. It is needed for the next round. So after the mitotic process, the cells grow and then go to the synthesis again. Now, how do the cells regulate the cellular reproduction, and how do checkpoints work? And what happens if something goes wrong here? And how would we as researchers and biologists recognize that? So as I said, there are two checkpoints. The G1S checkpoint, and in this checkpoint, we look for cell size, if it's correct, if it has enough nutrient, if it has the growth factors, and so in other words, it doesn't have enough energy, and if the DNA has been segregated correctly. And in the G2 checkpoint, we will also check those again, the cell size and the DNA replication mostly. Because at the G2, you can't proceed if there's any DNA replication issues. Now, the checkpoints, as you can see here, it says DNA damage and it says here DNA replication. This is because if you look here, division already happened, right? If division already happened and there was a mistake here that bypassed the system, then most probably that cell will carry out that damage, and it can be checked here in the G1S. So if there's any damage from the previous division, it will be checked again here. While during the synthesis and the gap2, this is more for the check on the DNA replication. Now, this happens because we have a set of proteins called cyclins, and as their name indicate, their level rise and fall. So these cyclin proteins are the proteins that regulate the progression of the cell cycle during the interface to mitosis. So there are three categories of these proteins: the cyclin themselves, so cyclin protein, and these are listed here as cyclin E, cyclin D, cyclin A, and cyclin B. And then you have the cyclin-dependent protein kinases. So cyclin-dependent protein kinases. So these are kinases that perform the phosphorylation function of the cyclins. And then the third category, these are the inhibitors of these cyclin-dependent protein kinases. So the inhibitors of this second category here. So now we have kinases. They are indicated as CDK, so cyclin-dependent protein kinases, CDKs. So you can see you have cyclin 4, 6, important for the G1. Cyclin 4 prefers D. Cyclin D binds to the CDK4, 6. Cyclin E binds more to the CDK2, as well as cyclin A, and then you have the CDK1. Now, there is a very important aspect that I will be describing in the next slide. When the cyclins are complexed with the kinases, then the cell cycle events are triggered. Okay? So you would need this first category, the cyclins, to bind to their kinases in order to allow the cell cycle to be continuing. Okay, now in this image here, you can see that between mitosis and G1, there is an exit. It's called G0. This is something we didn't describe in the previous slide because here we are discussing that, oh, the cell needs to be prepared for division. So it has to generate the RNA, proteins, and replicate the DNA. It has to be checked. Then, well, it passed the checkpoint. The cell is going to divide. Okay, the cell divided. It passed through all the steps. Now we have two daughter cells coming from that original one. And then these two daughter cells pass through the preparation phase gap one, where their cell size and DNA damage and other aspects are measured, and then they passed through the checkpoint. Now they are adult cells like this, and then the process continue again and again. Cool. But now there are some cells that don't go through this process all their life. Particularly speaking, I gave examples here like neurons, some types of stem cells and some like cardiac cells. These cells, they will exit the cell cycle, and cells can go to a pause where they don't commit to division anymore. These cells could stay there permanently as neurons and cardiomyocytes. They would not divide. And some of them could pause for a while temporarily, and then they can come back to the cell division. And obviously, this could be a very good way that the cells could regulate when to proceed in terms of the cell division. Now, this is a very regulated process. I'm not going to go to the molecular aspects of that one. Probably when we discuss senescence, I would give you more examples. But I just wanted to point out that some of the cells would exit the cell cycle either temporarily or permanently. And that's what we call the G0. Okay? But from this slide, I want you to understand that propagation of the cell cycle has to take place by a combinatory effect between cyclins and the CDKs, the kinases of these proteins. Now, in the next slide, I will describe it more. So how does that happen? Now, in this graph, you can tell that from the G1 phase to the synthesis phase to the G2 phase and mitosis, there is a very clear fluctuation of the cyclins. Okay? So in this slide, I will describe what we call cellular reproduction positive regulation. So in other words, how we push the cell or how we maintain that the cell goes into a division or a cell cycle. So what happens here is that cyclins and CDKs bind to each other, and then that's how they perform a function. In order to keep the checkup, the CDKs, which are the kinases, are stable. So they are not presented in this graph here, but their level is stable. So it could be a straight line like this. Okay. However, within the cell cycle, and as you can see in this graph, the level of the cyclins, they fluctuate. So some of these are upregulated and then downregulated. They are upregulated in the beginning of G1, like cyclin D, and it keeps on till the end of the process of the mitosis. Some of them are needed at particular steps like cyclin E, So it has to be upregulated in terms of the expression at the S phase, where the synthesis. So this cyclin, you can tell, it's responsible for the transition from the G1 to S. So as it's written here, cyclin E, it binds to the CDK2, and then it controls the transition from the gap into the S phase. If there is any issues in the process between the gap one into the synthesis phase, cell size problem, or what, and any other issues, DNA damage, then the process is gonna stop here. And you will see in the next few slides also more aspects that it links those processes together. Now, the levels of these four cyclins is fluctuating, and then you can see that there's a sharp drop just after the transition from one phase to another. So in other words, the cell produces this protein to make the checkpoint, and once that checkpoint is passed, we get rid of it. It directly gets degraded. And now there's another protein. In this case, it's cyclin A. And now cyclin A is responsible for the DNA replication in the S phase. So it's responsible for checking the process of the the DNA replication. Once that is checked, then it will drop. Therefore, the concentrations here of the cyclins with their kinases is very important and determine what happened in the cell cycle. Okay? Now, the process of stopping the cell cycle at certain point, it's much more complicated than that. This is just a snapshot of it, okay? I will describe how P53 causes transition to a cancer, and you will see how the cyclins are affected by that. Is this concept clear? Do you guys have any questions before I move on to the next slide? And if yes, please just turn on your mic. Okay. So to sum it up, cyclin and their dependent kinases will regulate these transitions. Now, let's say everything worked perfect. Then in terms of the process, these cyclins are generated and then degraded, and the cell cycle will proceed. Okay, how do a cell recognize that there's an issue? And if there is an issue, how can we pause the process? So in this condition, we would like to pause the process of a cell cycle at any of these phases. And in order to do that, we have proteins that are called the negative regulators. So the second group of cell cycle regulatory proteins, these are the negative regulators. These negative regulators, they regulate the active molecules that cause the cell cycle progression. So in other words, if you want to pause the cell cycle, definitely cyclins are your first target. Because as we said in the previous slide, the CDKs are stable. So their level are kind of stable. But the cyclins are the one that fluctuate. So you could imagine if you want to block the G1, you can simply block cyclin D. And if you want to block the transition of G1 to S, you will block cyclin E. Now, what are those proteins? There's one of these proteins that you already know. This is the retinoblastoma protein. Another example is, I give you 3 examples. There's many more. the p53, or sometimes called TP53, which is a tumor suppressor, and then the p21, which is a cyclin-dependent kinase inhibitor. So recall in this slide here, I told you that there are some cyclins, kinases for this cyclin, and inhibitor for the second group here. So p21 is an inhibitor for the cyclin-dependent kinases. So the p21 inhibit by those protein by binding to them and preventing them from activating their cyclin partners. Okay, so now you can imagine if you would like to block the cell cycle, it's binding of the CDK2, binding of p21 with the CDK2 will block the activation of cyclin E and A. So these two cyclins will be directly shut down because their function depends on the cyclin dependent kinases. If p21 binds to the CDKs, then the process is not going to take place. Okay, so let's continue first with the p53. It's one of the most famous studies genes in terms of cancer and cell cycle regulation and DNA damage. This is a multifunctional protein. Its major impact on the cell, it's whether it commits to the division or not. And if there is a DNA damage in the cells, then it has to go into the repair process before it can proceed. And if we jump into this part of the slide, if a cell is exposed to stress, and remember, we mentioned that stress doesn't mean all the time that the cell is going to die. Stress means the cell has to adapt. So as a response for that, the cells produce more levels of the p53. And p53 is a positive regulator of p21. So the more p53 you generate, the more p21 will be generated. And those two will accumulate in the cell. Now, when those two accumulate in the cell, it's less likely that the cell will enter to the S phase. And you can already tell why, because p21 is going to bind to the kinase that activate the cyclin E, right? And if p21 is elevated, then CD kinase is blocked, and then cyclin E cannot tell that the cell is ready to transition from the gap 1 into the S phase. So this can give the cell the time to fix any genetic damage before replication. Okay, so this is an adaptation process that could lead eventually to a disease. So let's go back here. Now, if there is a DNA damage that is detected, p53 directly blocks the cycle by the mechanism I just mentioned. Of course, there's many other enzymes that are recruited so that the DNA repair takes place. And at the same time, it gives this cell an adaptive process to take place. If the DNA cannot be repaired, then p53 should trigger cell death, apoptosis. So apoptosis is one form of organized regulated cell death. Okay, we will be describing types of cell death at the end of this lecture, at the beginning of next lecture. And this is forcing the cell to suicide in order to prevent the duplication of damaged chromosome or DNAs, like genes. Okay, so this is generally how the negative regulation work and the positive regulation works. So in the positive regulation, you have proteins that are taking place, taking care of all these steps. If we want to negatively impact the cell, well, all this has to be checked in order for the cell to proceed. And in case there is a damage, there is a system where can block these activators, cyclins and cyclin-dependent kinases, and therefore the cell will stop the division. So it can happen. In this example, we have two different aspects that are blocked, and they are mentioned here. Now, let's talk more about p53 and how p53 works. One might ask, okay, why the cells produce p53 if its accumulation could lead to a blockage of the cell cycle, right? Well, eventually the cell produce a very small amount of p53 because the system has to always check if there is DNA damage or not, and to be prepared to act very quickly. So in an unstressed state, the p53 that is generated, it becomes ubiquitinated. So this is a post-translational modification that leads to target that protein into proteasomal degradation. So most of the proteins that get ubiquitin in a certain sequence or at certain residues, those proteins are degraded. So any damaged proteins, in fact, in our cells could be degraded through the ubiquitination. And now this is a homeostatic process that most of the p53 generated in steady states, unstressed cells are ubiquitinated and then degraded. Okay. Now, why p53 is very famous? It's a tumor suppressor. It's mutated in almost 50% of human cancers. But what does it mean, that mutation? That mutation means probably the steady state is shifted to a disease state. So what happened in normal condition where p53 levels are basal, the transcription of the transcriptional control is under check, and the genes under the control of p53, like p21 and others, are minimal. What happens in conditions of a stressed state? Whatever that stress comes from, either if during the process of the replication there is damage in the DNA, or there's an exogenous factor like UV light or cancerogenic that cause damage in the cell at the level of the DNA, fragmentation, or there is a loss of the metabolic function. So there is a deficit in the oxygen or the energy ATP or the nutrients, or whether there is an expression of an oncogene, which is an opposite of a tumor suppressor. So this is a gene that positively induces cancer production. In all of these situations, we would require that p53 to block the process of the DNA replication and the cell cycle progression. So that p53 now, instead of being ubiquitinated, it will be phosphorylated and acetylated. And this will modify the stability of the p53 so that it can bind more stably to the DNA, and therefore induces those genes downstream of p53 more and more. I described in the previous slide the p21 function, and there's many other proteins that are downstream of the p53. You can easily tell that there's like more than like 500 genes downstream of p53. Here I listed some of them. They would control most of the diverse cellular functions. But in this slide, I will focus on only two aspects. The first aspect is that p53 induces apoptosis in non-transformed cell, mostly by direct transcription activation of the pro-apoptotic proteins. So what are the pro-apoptotic proteins? These are proteins that cause the cell, that force the cell to go to apoptotic cell death. Two of them, one of them called Puma and the other is Noxa. Okay, so I want you to remember these two names, Puma and Noxa. These are pro-apoptotic PH3 containing proteins. These are downstream targets of the p53. Combined loss of the p53 effectors of apoptosis, that this guy, Puma and Noxa, and the cell cycle arrest p21 that I described in the previous slide does not always cause tumor development. This is very important for you to understand that this is still an adaptive process. It's still, the cell is still trying to solve the issue and recover from that damage. Now, say this stress continues and the cell can't, then it will push more toward more Puma, more Noxa, and more P21. The more Puma and Noxa, this means more apoptotic cell death uh, arm of the adaptation. And the more P21, this means the cell will never pass the checkpoint where it continue to the continuation of the cell cycle progression. Okay? Somebody wrote something, maybe speed. Maybe. So there's a question here. So after cell damage is detected, the cell cycle is paused temporarily. After damage is fixed, does it restart? Yes. Or go to the next checkpoint since damage is now fixed. So I didn't get the question. So the pause in the cell cycle will not continue any step forward unless it is fixed. Okay. I don't know why this is coming back again. Okay. So this slide summarizes for you how p53 regulates negatively the cell cycle and how it can become now not anymore an adaptation process and leads to cancer. So if I go back here quickly and I simply give you a scenario where p53 phosphorylation doesn't take place. There is a mutation that causes at the site where a mutation in the p53 where the phosphorylation event doesn't take place anymore. Therefore, p53 is going to become more degraded and it cannot block the cell cycle. And it cannot generate Puma, Noxa, and P21. And therefore, the cell could skip this checkpoint and pass to the next one. Now, there are a few proteins that are mentioned here. You will see in the next slide, the checkpoint kinase 1 and 2. The checkpoint kinase 1 and 2, checkpoint kinase 1 and 2, these are upstream of p53. And these are, in fact, the enzymes that cause the p53 to be phosphorylated. So if there is a damage, in principle, some of these proteins that detect the DNA damage would relay the signal to the p53 in order to turn on this cell cycle adaptation process. Now, most of the mutations of the p53 are detected in cancer cells. These are point mutations. And these mutations cause a loss of the DNA binding of the p53 to the DNA. Okay, so it's very simple. A mutation that causes p53 not able to bind to the DNA, therefore the DNA can be no more activated by p53. And therefore, the downstream targets, Puma, Noxa, P21, cannot be generated. So this is a very easy explanation of a mutation in p53 that causes a point mutation in p53 that causes p53 not able to bind to that DNA and therefore not able to execute the transcriptional regulation. Therefore, these mutant p53 proteins are unable to regulate the transcription of the downstream gene, and that's a loss of function. Whenever a gene is not able to perform a certain function, we call it a loss of function. Okay? The other problem could be is that we will have a mix of the p53 that is the wild type that is able to perform the function and the mutant part. So they will start to compete because recall in this slide, we are saying that p53 makes tetramers, 4 different molecules, they attach to each other. So make a complex. So imagine 2 of them are phosphorylated, 2 others are not. Now we will have a dominant negative effect because these mutants will not exert the same function that the wild type proteins could do. And in other words, the more mutant you have, the more p53 that is not binding to the DNA. And therefore, this will play a role in the transcriptional regulation downstream of p53. Okay, so many of these tumors, they express different high levels of these mutant p53s. So, and this, in fact, could be how we can use this molecular tool to detect a cancer or to diagnose a cancer. In other words, you can easily make a probe that detects if this protein has been mutated. And the more that p53 is mutated is a very good correlation that could tell us that this person most probably will have, unfortunately, cancer. Okay, so I just hope this makes sense for you. And this here summarizes for you how the mutations of the p53 could lead to a loss of function, or if you want a shift from the homeostatic adaptation to a loss of function that could lead to a genetic material issues, like go forward without having the checkpoints, or having a p53 that is a mutant that cannot perform the function. So either by loss of the wild type functional activity, and therefore it doesn't bind to the DNA. So it's the loss of the wild type p53 transcriptional function, or it has a dominant negative effect because now you have the wild type and you have the mutant, and the mutant doesn't perform the function. So there's a like some sort of competition between the two. And then the third one is the gain of function, which is there is modulation of the downstream targets. Like imagine that these mutants will produce a positive feedback that could lead some of the tumor suppressors to be down or oncogenes to be upregulated. In this situation, there will be a gain of function. Because now, when you generate a mutant protein, that mutant protein could have a totally different function from the wild type one. So it can do a new, that's why we say it de novo gain of function. This means a function that normally p53 doesn't do, but when it's mutated, it's gonna do it. and therefore you don't know what is that. It could be a positive regulator. It could be like a block of the apoptotic process. It could be like induction of an oncogene, which can cause a disease. Okay? So these are the three different scenarios that could raise from a mutation at the level of the p53. And why we're discussing this now, because this is happening within the process of the cell cycle progression. So the cell is dividing, but this protein is very crucial in order to allow the cell cycle to proceed. And any changes or mutations could lead to those diseases that we discussed. So to summarize, this is another example where DNA damaging agents like chemicals, radiations, or cellular metabolism related products could lead to a damage in the DNA at the level of uh, the nitrogenous bases. Like when you say chemicals, radiations, and cellular metabolism, a chemical could modify, like we discussed carbon, hydrogen, oxygen, how they really interact. A chemical could bind to those nitrogenous bases and modify them. And therefore, the AT or CG binding to each other could be affected. And therefore, you could create either crosslinks or breaks, single or double stranded breaks in the DNA. So that's what exactly it says. Also cellular metabolism, because don't forget that DNA requires those nitrogenous bases, and the generation of the nitrogenous bases is in fact depending on the cellular metabolism. It has to have the energy to do that, and the nutrients, in fact. So now, if there is a lesion or a DNA damage that takes place, this DNA damage induces a response. It's called DNA damaged response. As I said, the best and the easiest scenario is to kill that cell. It will activate the apoptotic program and it causes the removal of that cell. However, the cells don't go always through that route because it will give a chance that this DNA damage response could, in fact, cause a pause in the process, but then eventually the cell can continue the cell cycle. So it will just make an arrest of the cell cycle for the moment so that it could be adapted, corrected, repaired, and then proceed within the cell cycle. Now, if it survived that unrepaired damage, then this causes the replication stress. And that replication stress means most probably that cell carried out genetic material that is not correct. Okay? And then it could survive with unrepaired damage. And that is what causes the genomic instability. Okay? So that we described kind of p53. We have p53 mutation. p53 mutation is carried out. Okay, if there's a mutant of that p53, then this process passes the check and it could lead all the way to the genomic instability. Now, say it went here and it is still able to go to from the adaptation process to the regular state or the normal state, then after the arrest, it can go back and continue because the DNA has been repaired, and then it can go back to a normal cell. Okay? So I kind of covered up and down, went back and forth, and I described the concept in different ways. And I want you guys probably to get exposed to BioRender. This is one of the figures that is made by BioRender. I don't know if you are aware of it. I think this is a software that you can use for generating images like this. Most probably it's free for many labs. I'm not sure if it's free for students. I would welcome that you really get exposed to this in the future because it helps you to illustrate the material very easily. Okay, this last slide, we will finish it before the break. it kind of summarizes everything I said, and it shows you more the loops, how it works. And I'm focusing only on the DNA damage here. In the next slide, I wrote for you what are these proteins. If you look, the name is complicated, but if you crack it down, it will be a kinase. So ATM is a serine threonine protein kinase. It's a master regulator of the DNA damage response. The same here. ATR is a serine threonine protein kinase. Again, it's a kinase protein that adds a phosphate. It functions as a sensor for the DNA damage and replication stress. Okay, so these two guys here, ATM, ATR, if there is a damage in the DNA, that damage will directly be detected by these two proteins. Okay, what do these proteins do? These are kinases. What do? They add a phosphate. They add a phosphate to what? To CHK1 and 2, which are the cell cycle checkpoint kinases, which are those cell cycle checkpoints here and here. Okay, so now we are having the full image where we have a DNA damage. DNA damage is sensed by two different proteins, and those two different proteins will activate the cell cycle checkpoint kinases 1 and 2. Now, the direct target of this kinase CHK1 and 2 is P53. Recall, P53 normally is not phosphorylated because it has to be degraded. But now there's the damage. The damage caused AT1, ATM, and ATR to be active, caused the CHK1 and 2 to become active. Those, in turn, they relate the signal to P53 to become phosphorylated, and therefore it's more stable. Great. P53 is more stable. This means it can make tetramers and bind to the DNA. Okay, so it binds to those DNA where it binds to P21 so that P21 can block the cell cycle quickly by binding to the CDKs. So that's one route. And P53 binds and activates PUMA and BAX, which are the apoptotic proteins. And this is where the cell has to decide either to later on leave the cell to continue if the stress has been relieved, or push the cell to die. Okay. Downstream of that, you can see other proteins that normally block the function of P53. So whenever there's this type of end, this is blocking the P53 because the signal doesn't need to stay forever. If everything went okay, then we need to block the activation of the P53. And this most of the time happens with the protein whip because the whip causes the dephosphorylation of the P53. So whip is gonna dephosphorylate P53, and that is enough to bring it back to normal and therefore stop the expression of P21 and PUMA PAX. And therefore the cell continue now in the cell cycle. Okay, we'll take a break for, let's say, 20 minutes. We meet back at 10. And the first thing I would like to do is to ask you if everything is okay at this slide before moving to the next part, okay? I'm gonna stop recording now and I will resume later on. Hello, are you guys back? Please turn on your mic. Oh, good. You raised your hand, Margaret. Thanks. Okay, first question is, how is it going so far? Is there anything that is not clear that you want me to repeat or focus on before moving for the next part? Because the next part is kind of easier and it's mostly like an application of what we have discussed here. Questions, comments? Okay. Okay, so let's move forward. So the second part of the second example of the negative regulation of the cell cycle, this we have discussed already a couple of times so far, which is the retinoblastoma protein. And as you can see, this protein is mostly depending on the phosphorylation event. If in normal conditions, it's very similar to the P53. In normal conditions, the retinoblastoma is not phosphorylated. And once it's not phosphorylated, the conformational change, it allows it to bind to the E2F, which is the transcription factor. And reminding you that these transcription factors, these are proteins that normally bind to the promoters of the genes in order to turn on the gene transcription. Now, if you recall in the checkpoints, the cell has to have some sort of check on the nutrients, the DNA quality, and the cell size. So if anything is related to a loss of a nutrient or the cell size, then retinoblastoma most probably will be activated. Now, in the inactive form, the non-phosphorylated state of the retinoblastoma binds to the transcription factor and it blocks its function. But when retinoblastoma is binding to that protein, it's blocking the process of the G to S transition. So again, as we described for the P53 and the cyclins, the G to S transition is blocked by the retinoblastoma. When the cell size when the cell size increase, suggesting that the cell is now ready to move to the next step, and therefore the retinoblastoma function has to be reverted, then retinoblastoma becomes phosphorylated. And this phosphorylation causes inactivation of it, or in other words, it leaves the E to F to bind to that DNA. Now, once retinoblastoma release those transcription factors, obviously you could think that the E to F will bind with many other proteins, and now the complex can start, and the process of generation of these proteins will take place, and the transition, the block on this G to S transition is relieved. And it's important for you to realize that we are just describing some of these checkpoints. There are others and many others that we can't really cover here, but it's important to know that a cell must pass all the checkpoints where all the positive regulators should be turned on, like the cyclins, and all the negative regulators, which are two examples I gave you, the P53 and the retinoblastoma, should be turned off so that the cell cycle continues and take place. If one of the positive regulators is not turned on, or if one of the negative regulators is always off or stays off, the cell cycle is not gonna proceed. Okay? One other aspect I want you to be careful for. Oh, let's see, what is it? This guy. Sorry, my screen froze. Okay, probably because of that. So one aspect I want you to be careful for is that also retinoblastoma is not only looking at the cell size, it's also correlated to the amount of nutrient that is available in the cell. Okay. So what happens that causes a cancer, and how do those cancer cells proceed to become immortal? Like I gave you two scenarios of P53 and retinoblastoma is if you go to the point where these cells, these two proteins didn't perform their function, or there's a mutation that doesn't allow the phosphorylation of these two proteins, then probably the checkpoints can be passed. And that's how cancer cells would bypass the checkpoints. But once they check these points, there is other steps that take place. In fact, the cancer cell become immortalized. And what does it mean, immortalization? It means that these cells eventually divide for unlimited number of time. So most cells of most cells of our body will eventually come to a point that they die, but the cancer cells, they are prepared to divide much, much longer, so they don't do that, even if they acquire certain mutations or DNA damages or changes in the genes. And with the every cell division, most of these cells, they have a protection that takes place. So what happens is that in cancer cells, there's an increase of the telomeres of our chromosomes. So normally, as cell divide, they lose part of these ends of the chromosomes that are telomeres. In cancer cells, they turn on an enzyme called telomerase, and that telomerase will most probably replace those lost parts of the telomeres. And therefore, the extremities of these chromosomes in which cause the cell at certain point to die in normal conditions, now they are protected against that thing, and then they will survive more in conditions of cancer. So there's a higher chance wherever there is a higher telomerase production for a longer telomeres, and therefore an immortalization of the cancerous cells. Now, there's another aspect you have to realize, that when these cells start to divide, and if the cell cycle regulation is disrupted, the normal situation is that there's a constant number of cells. That's why our bodies become to a certain point and then the cells differentiate. That's not something we see with cancer, because in normal conditions, our cell balance in terms of number of the cells produced and the number of cells died is balanced. But in the tumor conditions or cancer conditions, the proliferation index is much higher. And therefore, that's why there will be an outgrowth. That is because these cells are more protected, so they don't die. This is one condition. So, and this is, for example, one of the aspects we describe here, and with the mutations of P53 and retinoblastoma. Also, these cells, they don't activate the apoptotic process. So they are protected against the apoptotic process. So they have a double-edged effect that causes them to grow more and die less. Therefore, the growth of the cancer in the body is calculated by what we call the growth fraction, which means you can count the number of cells and how much they double. So the growth fraction is the ratio of the dividing cell to the resting cell. And in a tumor, this is very high. Normally, there is a lethal burden when we get to an index that is of around 1,000 cubic centimeter. That's when the cells almost reach 30 to 40 doubling. Okay, because the cells are going to start dividing and dividing and don't do any function. And this is what we call exponential growth. So the cells, they become 2, become 4, become 6, become 2, 4, 8, and such. Whenever we reach in a clinical setup tumors that are around 1 centimeter in diameter, this is mostly correlated to 30 to 40 doubling. And these 30 to 40 doubling correlate to a fact that the division of those cells going in an exponential condition. And that's how most of the time people would detect that they would have a tumor. So there's an outgrowth somewhere that is detected. I hope nobody gets that, God's sake. So with this, I end the part of discussion on the cell cycle itself and the regulations and the checkpoints and if it goes outrageous. So as I mentioned in the beginning of the class, that the best decision the cell could take is to die if there's a damage. And the worst situation would be, as I just described in the last slide, that the cell becomes cancerous. Okay, now let's move a little bit toward the discussion about the DNA replication, because all the GAP1 synthesis and GAP2 and the checkpoints and all that energy that we are spending during the interface is in preparation for the cell to divide. And in order to divide, we will have all the genetic material and the energy and the proteins, the machineries that are needed for the division to generate two daughter cells identical from that one that happens during the mitotic process. Okay, now if this is a somatic cell, it's mitosis, as I described before. If it is a gamete, it's going to be a meiosis. So before going to the process of mitosis and meiosis, I just want to describe a couple of DNA modifications. Okay. Before, we described the generation of the proteins, and then we described that, okay, one DNA sequence could lead to a messenger RNA that is transcribed and then to a protein. Then we described how RNA is spliced, so from one gene you can generate more than one protein. And then we generated from that protein different versions, one of them active and one is inactive by simply post-translational modification. The phosphorylation events that we described today on P53 and retinoblastoma are a good example of post-translational modifications of proteins that caused differential function. Now, in DNA, we have a similar aspect that takes place. So the DNA sequence, if we take a small part of a gene or a promoter of a gene that has a sequence, right? Now we can modify the DNA of that sequence by simply adding acetyl group or methyl group. These are the two most famous. Okay, so the DNA sequences can produce dramatically different phenotypes because of chemical modifications. So if we check the sequence of two different people at the promoter region and they have the exact same sequence, it doesn't mean the function is the same. Why? Because chemical modifications that we are talking about could alter the expression of that gene simply by modifying the nitrogenous bases. So you can have a sequence ATCGAT that is methylated or acetylated that could have a different function from the same one that is not methylated or acetylated. Okay, so an important example of this modification is the DNA methylation, which means you are adding a methyl group to the DNA. And this is an attachment of a methyl group to a specific DNA nitrogenous base, which is the cytosine, the C, okay? And normally this C is when it's followed by a guanine, which is the G of the DNA sequence, they form what we call the G-CRH region or the CPG islands. This means this is a region where you have a CG, 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 and then the C has a methyl group on it. Then we can call this as a GC-rich region or a CPG island. Okay. Now, how do these affect the function of a DNA? Whenever a CPG island is located near a gene where it becomes heavily methylated, then it's most likely that this gene is not going to be transcribed. So in other words, whenever you add a methyl group, there's always exceptions, okay? Generally accepted that DNA methylation causes the gene to become transcriptionally inactive, okay? But there are exceptions. For example, if we get a DNA from monozygotic twins, this means most of their DNA is kind of similar to like 99%, okay? Then there were studies that showed accumulation of methyl group on certain sequences of their DNA caused a change in their somatic appearance, and therefore they caused to have a different phenotype, which is a simple translation for what I just told you, that a sequence of a DNA, whether it's methylated or acetylated or not, could lead to a turn on or turn off of a gene, and therefore it will cause a change in the outcome of that gene and a change in the phenotypic difference. Okay? And this image here is showing you the example. So you have here a CPG island that is unmethylated. Then the gene is transcribed. Whenever the CPG island is methylated, the gene is turned off. And this has to do with the accessibility of that gene, because most probably when it's unmethylated, certain proteins can bind to that DNA and turn on the transcription. And when it's methylated, those transcription machinery proteins cannot anymore access to that DNA and bind, and therefore it will not turn the process of the gene transcription. So there is one thing that I missed to tell you many times. I have to go back to describe it. When we represent stuff in biology, sometimes we don't really take into consideration the size. So many times in those representations, this is a piece of DNA, right? And that is a protein. It's a transcription factor and retinoblastoma. For the schematic representation, this guy here looks bigger than DNA, okay? But this is in reality false. It's totally incorrect. The size of the DNA compared to protein is much like protein compared to DNA is much smaller. So please keep this in mind, because if you run, for example, protein and DNA on a gel, you can detect the DNA, but you cannot at all detect the protein together. You have to do it in a way that allows you to do that. So please be careful, because sometimes we do illustrations and we take it for granted, but it's not correct, okay? So now it goes the same here. It looks like the histone is by the same size of the DNA. This is not, of course, correct. Now, how do so this is the GC-rich regions, and then we will describe probably in a couple of slides the acetylation as well. Okay, but how do DNA get organized in our nuclei and get prepared for the division? So I probably you have done this before in molecular biology. It's just a refresher for you guys that the DNA molecules, in order to be packed, because they are very thin and long, then we need to kind of wrap them around certain proteins. These proteins are the histones, okay? And the histones there's of course more than one type. I'm not going into the details of that. And the DNA will generate these left-handed, so it's written left-handed spiral around these spools. So you will have the DNA left 100 spirals around those proteins, which makes what we call a combination of a DNA and a protein called a nucleosome. Okay, now how many base pairs between one nucleosome and another? What's the size of these histones? I don't really care about that because this is not the scope of what we are studying here. This is important in genetics probably, but not in this course right now. Now, these proteins and DNA called the nucleosome, they are bound together, and this will give us a structural organization of this sequence of DNA into what we call a nucleosome that can coil more to generate the chromatin. Okay, so it's a more condensed structure of the DNA that will eventually lead to the generation of the chromosomes as we know. Okay, nucleosomes are organized into chromatins, and these chromatins, as repeating units, they form what we call the chromosomes. Now, methylation could take place at the position 5 of the cytosine. It's presented for you here. And this is a footprint of what we call the epigenetic alteration. So the post-translational modification of a protein, we call post-translation modification. A post-translation modification of a DNA at the cytosine, presented here, we called epigenetic. Okay, so this is not a sequence-related change. It's a modification of the base pair by adding a methyl group or acetyl group. So when genes are expressed, chromatin is open or active. And as I said, it's related to the accessibility of those transcriptional proteins. However, when chromatin is condensed, and that's most probably because of the methylation process and histone modifications, these genes are inactivated. Okay, so now we have a piece of DNA that could be post-translation modified. So we moved from this simple sequence of DNA into a chromatin-like structure made of nucleosomes that could generate the chromosomes, and we have 23 of those, 21 somatic and the rest as gamete, and then 22 as somatic and one as the gametes, which is X or Y, sex-determining chromosomes. And then those are kind of wrapped in the nucleus in duplicates. So we have 23 pairs of total 46 chromosomes, right? Now, obviously, we are describing this as 23 because each one exists in duplicate. Now we have that cell in the nucleus. We have those chromosomes, and now we will discuss the process of the mitotic process. One last thing I want to describe before moving to the mitosis and meiosis is the DNA and protein interactions and what are the enzymes that can play a role in the process of activation and deactivation of DNA sequence. So there is a number of enzymes that contribute to the post-translational modification of DNA or the epigenetics. These are DNA methyltransferases. I don't know how much biochemistry you guys studied, but from the name, you can tell what the function of that protein. So it's a DNA methyltransferase. This means it will transfer or add a methyl to the DNA, where most probably is going to be the cytosine. There is another one called histone deacetylase. So this will remove deacetylase. So it's a removal of acetyl, an enzyme that removes the acetyl group from a histone protein. So a removal of acetyl. So then there is a histone methyltransferase. So this is adding a methyl to the histone protein. Okay. Now, of course, there are other complexes that take place when there is a generation of transcription or replication. Those proteins, they don't function separately. They form complexes. And some of those proteins, like complex nucleosomal remodeling factors, could come into the show where they can perform multiple changes on the complex itself. It could be across different proteins. It could be of one of the proteins in the complex. So histone modification and promoter methylation or acetylation, like this is one of the examples, could regulate the gene expression. Okay, and methylation on the DNA is mostly downregulation of that expression. Now, genes that are transcribed when the chromatin is modified by the addition of acetyl group, and these are added to the lysine groups of the histones. So we could add like acetylated group to the histones, and there is a domain where people are studying all these epigenetic modifications upstream of genes, what an acetylation on each of these lysines could do. This is a very, very uh, well-known domain of study, and it's going like really, really deep on that. So generally speaking, turning on a gene, it could be performed by a reverse function. So gene expression can be turned off when an acetyl group is removed. Okay, so these histone deacetylases, in other words, perform the same function of methyltransferases. Similar function, I want to say. Not the same function, but a similar function. Because a gene is turned on because it's acetylated. Now you remove the acetyl group, you can turn it off. In other words, either this or when you have a GC-rich region, those genes will be methylated, right? The DNA is methylated, and that's another function where, another aspect where the gene expression is turned off. Okay, how can we use that? It's very simple. We could detect histone methylation on certain of the cytosines or the deacetylation as markers of activation of a gene. So there's tons of research that showed that certain genes are turned on or off simply by activating histone deacetylases or methyltransferases. So this is a kinda expressed here. So you have an active chromatin in this situation on the chromosome, on nucleosomes, where this DNA is wrapped around the protein. Again, this is not really reflecting the reality, where the transcription factor now is going to be able to bind to that DNA because it is acetylated. Okay? And the acetylation process with the binding of, the binding of the DNA with the transcription factor will turn on the gene transcription generation of messenger RNA and a protein. Now, what I just told you is that if we deacetylase that histone, so we remove the acetyl group from the histone proteins, then most probably that transcription factor cannot bind. Or if we add methyl group, then most probably that transcription factor is not going to be able to bind. And in both situations, if the transcription factor didn't bind, then the transcription process is not going to be turned on, and therefore there will be no messenger RNA and no protein. In other words, the transcription is inhibited. And that's what we call inactive chromatin. Okay? So these are kind of the post-translation modifications on the DNA and on the proteins that are affecting the DNA protein interaction and therefore transcription of genes. Okay, Why is this important? This is important because obviously all the cellular functions and that are downstream of gene transcription and translation affect the cellular function, including the replication process, because recall that the process of replication depends on proteins. Yes, the genetic material is being copied, but who is doing that? These are enzymes, and the enzymes, these are proteins. Of course, we will need the genetic material to be correct in order to be copied, to generate the two copies from that DNA sequence. And just to get you an idea, there were three different models suggested that the DNA is replicated. This is called semi-conservative. This means each parental is being copied into a newly generated helix. So if we take the double helix of the DNA, each one is being copied into a newly duplex, and that's called semi-conservative. There were many others, like two others, I want to say, that turned out to be not correct. And again, we are describing the eukaryotic replication of DNA here. From this, if I go back one step there, so we had this DNA sequence, nucleosomes, chromatins, and then the chromosomes. And those chromosomes now, we are moving there, that chromosome, if we go backward, it's here where the nucleosomes are, and then we are getting back to the single nucleotide sequences here. This is one helix, and this is another helix. And it's bound by all these machinery, transcriptional machinery proteins. So we would need first free nucleotides, which is A, T, C, and Gs. So the energy of the cell has to be enough in order to generate those four different nucleotides. That's why a checkpoint detects the amount of energy the cell has, because if we don't have the basic blocks of the protein of the DNA molecule, how would we be able to generate two copies of that DNA? And therefore, it's important to check that before proceeding. And that's why probably cells will stop dividing where there's no nutrients. Then you would need all these enzymes that are listed here, and we will be discussing in a couple of slides down the road. You will have a DNA polymerase, which is a protein that polymerizes a newly generated DNA strand by reading the original one. So this is the DNA polymerase sitting here and recruiting this nucleotide into the strand. Now, as you know, if we take this continuous, so we start replicating this protein, this DNA from here, and we start reading and generating a double helix, it will be continuing all the way till the end of the chromosome. That's on one side. And that's what we call the leading strand because it's a continuous generation. On the other side, you can't continue because all the time you have to recruit the polymerases and generate fragments that are linked together. So there's only one that is generated here. It's this guy. Imagine after 10 like bases we reach here, we have another strand. And those, we call it the lagging strand. There's another step that needs to be done where we ligate those together. Okay, so we have DNA polymerase that goes this way on the leading strand all the way, and there's many DNA polymerase. One of them bind here and go all the way, and then another one bind and go from here to there, and then there will be ligation of those molecules. Okay, and you can see there's an enzyme here called helicase. This helicase, as its shape indicates, it is gonna unwind that DNA molecule because normally DNA molecule is just like compact spiral shape like this, where the two helices are bound to each other, like as you see there. So this enzyme is kind of clearing the way for the polymerase and the other polymerase to bind and therefore perform the replication process. Of course, this machinery is very simplified here, but the DNA polymerase is a complex. There are multiple polymerases. Some of them perform the function of adding a nucleotide into corresponding to the complementary strand. And of course, some of them play a role in the DNA, how it's called, proofreading, to make sure that, yes, this was a T, we put an A across. And now this is a C, we put a G. Otherwise, the process will stop here. And that's how this machinery can regulate or relate a signal of DNA damage to the cell cycle that I described before, and then activate those cell cycle DNA damaged proteins, and then activate the p53 and then block this process. Okay, so altogether, this is called the replication fork. And there are other proteins to be added. Okay. So many enzymes, as I said, are involved in the DNA replication. The DNA-dependent DNA polymerase. So this DNA needs information from a DNA in order to generate another copy. So it polymerizes a new copy of that DNA depending on the sequence of that DNA. It polymerizes, catalyzes, and regulates the process. A DNA needs the support of other enzymes. So if we look at the DNA polymerases, there's 3 of them, polymerase 1, 2, and 3. And some of them, they have different activity. So for example, polymerase 1 has a DNA repair activity. It could polymerize from the 5' to 3'. It could exonuclease. So polymerase meaning adding the group, adding the nucleotide. It has a 5' and to 3' and 3' to 5' exonuclease activity, because as it's moving and it's adding that guanine, say it make a mistake here. It took, let's say, an adenine and put it there. It has the ability, an exonuclease activity, where it could stop and remove that out and then add the right correct one, which is the guanine again. Now, DNA polymerase 2 is responsible for the primer extension and proofreading. So as we said, there is this enzyme is going to check if the enzyme, if the polymerase 1 did the right job. So it's going to keep a check if everything has taken place correctly. And the polymerase 3, it's responsible for all the in vivo replication of the DNA. So this complex is like all taken together. Again, please remember that when we describe those things, we kind of dismantle them and give them function. But remember that this is a process that is taking all together in the cell, and all of them are involved within the same time. Okay, so I added for you here the names of those enzymes and what their function is. I don't really want to go to all the details of what each one does. Probably I'll let you read through it, and if there's anything not clear, please come back to me. Like we described helicase, we described ligase, because as I said, ligase has an important function, because say that this lagging strand starts polymerization here, it has to go that way. The problem is the fork and the replication fork is moving that way. Therefore, there will be an empty spot here that this polymerase cannot go back. It has to go always from 5' to 3' end, which means another transcriptional machinery, sorry, another DNA polymerase has to bind somewhere here when it becomes here. And it moves and generate the double strand. Now you have two molecules of DNA that you need to ligate together, and that's why you have the ligase. And this is the enzyme that joins together the Okazaki fragment. And the Okazaki fragment, this is one of them, and there will be another one here, and there will be another one already probably generated there. So you will have many Okazaki fragment that is generated on the lagging strand. And as you can say, Okazaki is the scientist who found out those um, oh my God, replication uh, machineries. Okay, there's also primase, endonuclease, and the single-stranded DNA binding proteins. So probably this come in the next, or no? Wait. Okay, no, they are not there. So there is the primase, it's the enzyme that helps those polymerases that bind on the first site to start the replication. The endonucleases, these are needed for the correction. These are exonucleases or endonucleases. Endo, this means it cuts within the DNA molecule. Exo means removal of the last part of that DNA molecule. And then the single-stranded DNA. Once you generate that fork by the helicase, you need some other proteins to maintain this fork open. And that's the function of the single-stranded DNA proteins, because they bind and they don't let that helix to wind again till the replication process take place. Okay, now, similar to what we discussed in the transcription where RNA molecule is generated and it's also following 5' to 3' end directionality, and the addition of that nucleotide, it comes when it has the 3 phosphates. It's exactly the same here, but now we are talking about DNA. So we got this DNA being replicated, so it's going that direction. So this means Okazaki fragments will be generated on this direction here, and we are adding more of this nucleotide complementary to this parental molecule, and the others here complementary to this parental molecule. Now, if you see, it's always growing from the 5' to 3' end, okay? And the way it happens is you get, for example, here, it gives you an example that it already did the first, the second, the third. The fourth is going to be a T on the parental strand or the template strand. Therefore, we have to have an adenosine, so it's an A. Now, on that sugar group, we already have at the 5' end 3 phosphates. What we will do, exactly as we described before, we will lose 2 of these phosphates out, and these phosphates are not lost. In fact, these will give us a lot of energy, if you recall the ATP, ADP. And then there will be a generation of the phosphodiester bond between the 3' on one and the 5' on the other. And the process will carry on all the way. So that's how it takes place in the RNA. That's how it takes place in the protein. There's not really in the DNA. There's nothing really different except that the ribose between the two molecules. Okay, so now we can say that within the interface, we prepared everything needed for that cell to divide. So we generated all the protein, we make a checkpoint that we have enough energy, we have enough organelles, we have enough DNA. And then we generated a copy of that DNA, and then we prepared that cell to grow to a level that it can divide into 2 and generate the 2 daughter cells from the same one. So all this process now is ready. We checked that protein, that the replication process is okay. We checked that there's no DNA damage. and now all the checkpoints are turned off. The positive checkpoints are turned on, and the negative regulators are turned off, so everything is clear to proceed with the mitotic process. And now the cell is going to go through the process of the division, and it will pass it through the 4 different phases that you guys know by heart, I guess. So the cell has to be prepared to become 2 daughter cells. Now, I want you to remember a few things, and probably I go through some of the terms that we will say as we describe the process of mitosis and meiosis. So I want to make it clear that these somatic cells containing 46 chromosomes, they are 23 pairs, so that's a duplicate amount of chromosomes. With exception, it's an XX or an XY regarding the sex chromosomes. Okay, now the gametes, these have only 23 chromosomes. Okay, because the gametes are a transitional state of life, I want to say. The gametes are generated in males or females as a sperm or eggs, right? And they are haploids, so 23 chromosomes, so they can't survive on their own. In order to continue their life cycle, the 2 gametes has to meet together through the fertilization process, where sperm, egg meet together, and then you have 23 chromosomes that come from each gamete to restore the total number of chromosomes that could generate an embryo. Now, you can do this by in vitro fertilization these days. Mitosis is the cell division of diploid cells of the 46 chromosomes in the somatic cells. Meiosis is the formation of the haploid cells from those diploid gametes. Sorry, sex cells. Okay, now autosomes, for those who don't know it or didn't study genetics, autosomes are the 22 pair of chromosomes in males and females are exactly the same. They perform different functions, not related to sex. However, sex chromosomes are only 1 pair, the XX or the XY, and then the size of the X chromosome compared to the Y chromosome is different. Okay, which gives you an overview back that, okay, now we have the DNA that could be methylated, acetylated, or histone, wrapped around histone that could be methylated, acetylated. It's wrapped, you have a coil of a chromosome, you have that chromosome. Now we know what type of chromosomes we have, and let's proceed in dividing those chromosomes. Now, homologous chromosomes, meaning they have a similar sequence, right? And when the cell wants to divide, it has to generate what we call a duplicate of those chromosomes. Now, if we look here, these are homologous chromosomes. They have a kinetic core where they are attached to each other. When you want to replicate those, you have to add a copy of it. So you add 1 copy of that chromosome, so it becomes 2, right? And this guy becomes 2. So that when we will do the mitosis process of these somatic cells, it will be very easy. We can take this part into 1 cell and this part to another cell, and therefore now you have the genetic material easily divided between 2 daughter cells. Okay, so I don't know if you guys have already studied before mitosis and meiosis. I'm not gonna go into deep details. It's described here, and I added for you a very nice video that you can watch at your pace whenever you want. But it's important to know that there are at least two categories that I want to describe in terms of the separation of the sister chromatid: the disjunction and the non-disjunction. So when we say disjunction, this means there's a normal separation of the sister chromatid during the cell division. When we say a non-disjunction, this means an error had happened where the homologous chromosome on sister chromatid failed to separate. Because in this process here, we generated a copy of that in order to 100% give one cell a copy of each. So we have two chromatids, each one will go to one cell. But if for certain reason we didn't really give all that part of that chromosome, this is a non-disjunctional division, which could cause a change in the number of the chromosomes. Because imagine one cell gets those two copies. So automatically, like instead of this blue chromosome, normally it's one of them go to one cell and the other is to the other daughter cell. If both of them went to one cell, automatically that cell has one chromosome more and the other daughter cell has one chromosome less. Okay, so this would cause what we call aneuploidy, which means the number of the chromosome in that cell is not 46 chromosomes in a human. And this causes, of course, genetic diseases. So in mitosis, we have, I go back here, you have that chromosome that becomes duplicated. And once it's duplicated through the prophase, it will be aligned and through the metaphase, and then it will be separated into two daughter cells. Each one has a 2N. Okay, so that's a somatic division. The process of meiosis is a little bit more complicated because most of the genetic modifications that take place at the time of producing a new offsprings has to happen before the gamete is generated. And that's why meiosis is divided into two parts, meiosis 1 and meiosis 2. Now, meiosis 2 is very similar to mitosis, where you have chromosomes and then you can divide those into the offsprings, where n equal 1 become it, like one cell becomes 2. But the only difference is that it has only n equal 1, not n equal 2. Remember, as I said, the gametes has only 23 chromosomes, not 46 chromosomes. Okay, so now you will have to first align in the meiosis 1, you will first have to align the chromosomes that are homologous. So chromosome number 1, we have two copies, we have to align it to the other one. So that's what we call the tetrad align at the metaphase plate. So we have chromosome 1 and it's chromosome 1 and chromosome 2 and chromosome 2. And then there should be this process, what we call homologous recombination, where these chromosomes will exchange the material. And then after exchanging the material, you can see that there is a chromosome in red and one in yellow, and another one in red and in yellow. And then you can see that you got the yellow chromosome, which is the second one of the pair, if we call a pair of two, we got the yellow one with a small piece that comes from the red into one cell. And the red one with a small piece from the yellow one, as you can see here, into another cell. So the two chromosomes, they sit like face to face, they exchange material, and then chromosome number one goes to cell number one, chromosome number 1A go to the other cell with the exchange material. And then the next step is repeat what we have done here, similar to meiosis. We separate the two chromosomes back, but now we will have only one half of those number of chromosomes. And these, during the end of the meiosis 2, we will have 4 different cells from the original in meiosis. And each one of this has only 23 chromosomes, ready to be, like if this is a sperm, it will be fertilizing the egg in order to get back to the 46 chromosomes and produce an embryo. And here there's a description for you for all the steps, what happened in the mitosis. And now we will move to describing some aspects of the genetic diseases. I know this is already heavy, it's already 11. I will go only for 5 minutes more or 10 minutes more, and we're gonna stop. And I will let you go through the rest of the material. There's nothing really very difficult in the coming slides, because if you got the concepts that we described so far, these are just an application or an example. So a genetic disease can be caused by a cause, which is described before as a mutation, or a combination of variations in the DNA. That imagine you have changes in happened across the two chromosomes here, and one of the chromosomes carried more compared to another. That's already a genetic related disease. But broadly, we can divide the genetic disease into 3 or 4. The fourth one is going to be a mix between the metabolic diseases and genetic diseases. The third one is going to be multifactorial, where you have a genetic disease that could cause a heart disease, for example. In other words, a genetic mutation that causes a heart disease, or a mutation that causes cancer, a mutation causes diabetes. This is a multifactorial related disease. But the two that I'm describing today is the monogenic diseases, which is a single gene that causes a single disorder. And these are mostly described as Mendelian disorders. And the other one would be a chromosomal aberration. This could be a change in the number of the chromosomes or the composition, meaning having more DNA translocated or inverted, or the sequence has been changed. But it's more than one single mutation, okay? Now, why this happens, I described before that it could be a genetic, it could be inherited, but it could be also environmental factor and exposition to compounds or UV light or diet or stuff like that, like radiation. Okay, so the 3 different types of genetic diseases, these are single, the monogenic diseases. These are alterations in a single gene that could lead to a disease. What are the biological mutagens? These are viruses. As I said, virus could come insert its DNA and it could cause a mutation that is added to some of our genes. Transposons, these are in fact parts of our chromosomes that exchange places across the genome. It's one of the aspects of the genetic stability, in fact. Chemical mutagens like smoking, heavy metals, pesticides, alkalizing agents like ethylene bromide and such, UV light, radiation, ionizing radiations, and gamma rays, all that could cause changes in your DNA and could cause a single mutation. Now, a single mutation that it changes an amino acid. Like in this condition, it's serine to aspartic acid, or aspartic, I think so. Then this is called a missense mutation. It's very simple. The amino acids before and after that single mutation, they still exactly the same. It's only just one change in one amino acid. That's called a missense mutation. Okay? But still, imagine it could be like silent. It could not cause it wouldn't really cause a disease, but it could be very dramatic. Imagine that this site here, the serine, is the site where p53 is phosphorylated or retinoblastoma is being phosphorylated. So it can be really dramatic. Okay, so a missense mutation could cause a disease or not. The second one is the nonsense mutation. The nonsense mutation is also a single mutation, but now you are simply introducing a stop codon. So you are truncating the protein. So imagine now you're having alanine, isoleucine, serine, tyrosine, and it should continue to phenylalanine. However, the tyrosine has been replaced by a stop, so you truncated the polypeptide. And the third one would be a frame shift mutation. And now the frame shift mutation is not only one single change, it's more than one. You need at least two. Okay? So what has happened here, you inserted two different nucleotides. You started with alanine, isoleucine, and now you shifted from serine into glycine. So everything downstream isoleucine is different. So that's what we call frame shift mutation. So the whole frame has been shifted just after that mutation. So the protein that is generated downstream, the polypeptide, is totally different from the original one. So this is frame shift mutation. And all those could cause diseases, except if we are very lucky that a missense mutation is silent, it doesn't really cause, like, for example, this aspartic acid is not, aspartate is not phosphorylated, or it doesn't really affect the function too much. But if the question is, do all mutations cause disease? The answer is no. Some of these are silent. In most cases, yes, they cause, because here I can give you two scenarios. One scenario is that the protein is being generated, and another one, the protein is being truncated. But sometimes truncated protein could perform the function, so there will be no disease. Some others, the truncated protein, or the short version of the protein, truncated means short version of the protein. In this situation, it doesn't do the function, then it can cause a disease. The most dramatic would be the frame shift, because this is a totally new protein, and that is most of the time causing diseases. A long list here, many of them we passed on. I'm not gonna go through that. And I'll let you guys stop here and read through the rest on your own. And in the next, I will come back to describe more these few slides if needed. Okay, so please go through these slides on your pace. This is just an explanation. For example, here we are describing that there's an addition of a chromosome like Down syndrome or Edwards syndrome. In this one here, we are describing that there is an addition of one sex chromosome, either X or Y, or a loss of the chromosome. Here we are describing a translocation where a small piece of the DNA has been translocated to another piece. And then, of course, this will cause a change in the gene transcription, inversion and translocation, so within the chromosome or across different chromosome. And finally, certain aspects on the fact that we have only limited number of genes on the Y chromosomes, and some of these genes are only on the X chromosome, and sometimes we have a more vulnerability that these genes are turned on or off on X or Y chromosome. So what I'm describing for you as an example here is the fragile X chromosome syndrome, which most probably you know that it will affect males more because they have only one copy of the X chromosome. So thus such diseases could affect the males more than the females. And finally, the factorial diseases. We will come back to these, of course, like heart disease, genes affecting the basal function of the cardiac muscle caused by a single mutation. Rheumatoid arthritis is one of those because we have a mutation in the HLA gene that could lead to a chronic disease. So please go through these slides. They are just application, and I just described them many times as I described the cell cycle, the mitosis, meiosis. It's just an application. And we'll stop here. So as I said, just a couple of things. Today afternoon, I have a meeting. We still have issues on my Brightspace. I have no idea why this year it's like that. So I am dividing you into groups for the assignment. The groups will be 2 people per group. You can assign yourself. You cannot assign more than 2 people in a group. If you can't find a partner, we will discuss this by next week. If you decide to do the assignment on your own, you are welcome to do that. The assignment will be about a rare disease where now that we described all the molecular aspects of genetic diseases and proteins and transcriptional translation and post-translation modifications, you can easily describe that. It will be the rubric will be added today this afternoon, and the groups will be there. So please, once you get the announcement, go ahead and choose the disease you want to work at and choose your partner. The best way you communicate with your partners before, so if you're going to group 1, you tell your friend or colleague that you want him or her to be in that group. Then next week, I will make sure before the class starts that everybody has a group and everybody has been assigned to a disease and you got the rubric. After that, you have 4 weeks to get that done. And I will get you in touch with the TAs who are going to be responsible for the correction process. On Friday, I will be released the first survey. It is 2 component, one component that is not graded and one component that is graded, but both of them are mandatory. The first survey is going to cover 3 to 4 points. It's going to be unproctored and you have 5 days to do it. So in other words, you are getting bonus on this already. If you have questions, please go to the discussion forum and add them there. And I will be more than happy to meet you in the office hours if needed. And with this, I wish you a good week and hopefully a good semester and a good day.
Yes,
Anybody?
you too, sir.
Yeah, if anybody has a question today, I will stay for a few minutes and I will stop recording right now.
Thank you.